We would have different
What?
primers. That's what they're called. Yeah. So there are different primers that you use to extract the like depending on what species you're targeting for, you would have a specific primer that you would use to extract that DNA. For this project specifically, we're looking at invertebrates. I wish I could go into more details about it, but unfortunately we don't extract the DNA here. We send them to our collaborators at Stanford University and they do it there. But as far as I know, they use like a specific primer for vertebrates to extract the DNA.
Push in here.
I don't see any animals.
No, just seemed very out of place then.
Yeah. Maybe tumble down.
Yeah, thanks, Jake.
Yep, all right. Come on.
Yeah, and thanks, Mike Connie, for that explanation. And yeah, I just want to take a second here and just acknowledge the tremendous support that has been provided in the planning and now in the execution of these expeditions by the wonderful staff at the Palau International Coral Reef Center. We've been working together for close to 2 years now in planning this expedition, and really appreciate the support, helping with logistics, helping with permitting, helping with a whole bunch, and it's just so wonderful to have various folks from Pickrick on board, and for me personally, very rewarding to share this watch with Imade and with Maikane. Having that local expertise on board is just wonderful. So big thank you.
So another follow-up on the eDNA. How many samples have you collected so far?
So far we've collected 5 surface samples and 5, or yes, no, today is going to be 5 ROV samples. So 10 in total. For the ROV samples, we trigger 6 Niskin bottles, and each bottle you come up with 3 replicates. So 3 times 6, like 18 for each ROV dive. And yeah, for the surface sample, we have 5 Niskins, so we have 15 replicates.
So would that be 165 replicates or something like that?
Yeah.
It's quite a lot.
It is a lot.
It's not just 5.
And the thing is, like, we
trick of light?
have 250
Looks like
ml
something.
cups,
Right.
and and for each filter you have to filter a liter. So I was doing the math with Pierre the other day, and I'm like, so basically we're doing 60 pours
Yeah.
with all 6 of these Niskins.
Yeah, it's a lot.
It's an arm workout, yeah.
Definitely.
Yeah.
Yeah.
Push in there. Oh, maybe it is just sediment.
It's a xenophyrophore.
Oh.
Yeah.
Oh, I love these. This one looks quite different than the other
Is
ones
this
I've
single
seen.
cell?
Yep.
It's a
organism?
4M.
Giant protist. Cool. I just looked, like I saw the way it kind of looked like it was casting a shadow. So it was kind of a weird texture.
Yeah, Jake, if I can do one more, try to get a good shot with the cinema cam.
Yeah.
Okay to zoom in?
Let me push forward a little bit. All right, there you go. Oh, I might have dusted it. You can, yeah, you're okay to zoom in. All right.
Thanks, Jake.
Yep.
We have a lot of viewers interested in the whole eDNA. So is the DNA composed of bits of things like the shedding exoskeleton or feces or?
Yeah, so I think fish and other marine organisms are pretty similar to us humans in how we shed skin and hair. They too also shed their skin and
So
scales
fish-
and whatnot. So the eDNA samples that we collect could be anything from feces to urine and shedded scales, and like little bits of, or like if a shark ate some big fish and it'd be dead
Push in
and
there.
it had some blood, it would also be in there. Yeah.
This looks like a Bathytyphlops. It's a blind lizardfish.
Nice. It's cool.
Oh, they're ambush
Whoa.
predators, so they sit on the seafloor and just sort of wait, and they can move very, very quickly for short periods of time. I believe we suctioned one of those a couple dives ago.
Yeah, I think so too. Looks really similar to what we have.
What's in the Atalanta camera?
Cuskeel?
Oh yeah, that's another Acanthonis
Oh.
armatus. They are very abundant here, which is unusual. Usually we see like one a dive, but we've been seeing them consistently.
Again, for eDNA, how do you collect surface samples and at what depth?
So we collect the samples using a Niskin bottle. It's pretty similar to the ones we used to collect with the ROV. Basically, it's this big cylinder with like 2 holes on each end, and there's like a little so I really don't know how to describe it. I could probably like post a video of us collecting one of these days. But yeah, we drop it down to like right below the surface, so like a meter below surface, and then we send the messenger, which is like this really heavy thing, and it triggers the
Caps.
caps,
On
and then
the
it
top and bottom,
closes,
yeah.
and then the water stays
Yeah.
in the cylinder. You know what? I think I'll make a video of us collecting the
Yeah, that'd be
samples.
great.
Yeah.
Yeah.
This sounds like an Instagram takeover to me.
Mhm.
I just might take over the Instagram account for a day just to do eDNA stuff.
That would be cool. Pretty soon, in about 80 meters, you'll see an eDNA collection with the ROV, which instead of using that messenger weight, we have these little colored balls that
There is something
are
on the
pulled
sediment off to the right.
trigger those caps closing for the Niskins on the ROV.
Oh, it's the Bathytyphlops. It's probably the same one that we just sort of startled. It's fun to watch it swim.
Yeah.
Seems like it takes a lot of energy though. You know, you see all the other
Alrighty.
eel-like fishes that it seems a lot easier for them. But
Looks a little less efficient.
yeah, not as efficient of a swimmer.
Zumba.
It can do short bursts
for a
really quick,
push in
but
there?
long distances, not so much. Yeah.
The way he's swimming, it looks like he pulled a hammy when we startled him last time.
Yeah, they get tired.
Try to line up one more shot with Cinema Cam.
Yep.
Maybe even brighten it up a little bit. Yeah. Nice.
That's great, thanks.
Yep. All right. Come on. Thanks.
It's another one of these skidded down rocks.
So for scientists ashore that are following from shore, we're midway between waypoint 2 and waypoint 3. And we have about 8 hours of bottom time to go. So we'll try to cover some ground here, so we'll have to climb another 370 meters to the summit of this feature. So this seamount has an interesting kind of ridge-like feature at the top, and we're hoping to make it to that top. Now, seamounts have been known for a long time to be these hotspots of biodiversity. So work done in the 80s where a series of current meters had been put on different parts of the seamount, and basically figured out that on the flanks of seamount you have higher currents, and you have these circular features, circulation features on top of the seamount known as Taylor columns, and they trap nutrients and provide favorable conditions for a whole bunch of suspension and filter feeding organisms that are typically found towards the flanks of seamounts, are also at the top. And when we're designing these dive tracks, we're purposely trying to cover a wide range of depths and of habitats to try to get a holistic view of what kind of habitats, environments, and species might be found on them. So yeah, people that are not from Palau but probably have heard from Palau because it is really, you know, it's close to the coral triangle, the place on Earth where you have the highest biodiversity of marine organisms. Palau, we're very close to it, so now we have several hundred species of corals, on the order of thousands of orders of fishes. That's just in shallow water, so the shallow water ecosystems have been relatively well studied. In deep water, we're still in our infancy, we're still learning what's down here. And so these expeditions are trying to put a little dent into what we know on the diversity of this great treasure that is Palau's oceans. Just take a quick snap zoom there. Another one of those
let's get a
hermit
quick zoom
crabs.
there.
Yep.
Yeah.
All right.
We've been seeing a lot of these.
Yeah, this is the third one.
It's just, if it's okay, I'll like to try.
I feel like everyone we've seen has one more polyp on its zoanthid backpack.
That's great. Thanks, Jake.
Yep. Fish.
There's a fish to the port. Ooh, there's another lizardfish. We gotta catch them all today.
Pushing there.
This is Bathysaurus mollis.
If it's
Again,
okay,
a
I'll
deep sea lizard fish.
try
Again,
to
a
zoom in with the cinema
ambush
cam too.
predator.
Yep. Go ahead.
So do the lizard fish have any bigger predators?
I don't believe they have any. Well, at least
Check.
not this
Yep.
fish does not have any predators down here. No,
All right. Come on.
this fish is the predator.
Okay.
The ipnops, the smaller one, could be eaten by another larger fish.
Jeff just chimed in and said that this fish actually bites the tails of rat tails and cusk eels, so it is the top predator indeed.
Yeah.
And he said that he's seen them attach fish and shrimp, attack fish and shrimp of the same size.
I bet they would. They're pretty ambitious.
Another one of these sediment slumps.
Yeah, this looks like the type of like mudstone that we would see on the continental shelf in like a canyon area.
Yeah, definitely. A very landslide kind of exposed. Seems like very steep. Do you have a slope map on iPack?
No, I don't have a slope map here.
That's okay. We can kind of get the sense.
Well, with the contour lines being as close as they are, it's decently steep.
Yeah.
I want to say probably 20 degrees, 25. Oh, there's a fish.
Cuskeel?
Mm-hmm.
Watch out for that lizard fish.
Right? Looks like it's probably another acanthonis.
You got a quick zoom there?
Yeah, that big head is a giveaway, but then you zoom in, you see the little beady guy and the little, like,
Jake, okay to do
cute
a
little
zoom in
snout.
with
Yep, go ahead. Yep.
Thank you.
Yep.
That's great.
All right. Fun.
Yeah, this has been another very fishy dive.
The fish like these sediment areas. They're good foraging areas.
It's a nice view in Argus at Atlanta. You can see the edge of that geologic kind of slump.
Yeah, and that's a good reminder for those viewers who are watching us on our website. You can toggle between 3 different cameras, so definitely encourage our viewers to take advantage of that. Sometimes you lose the sense of scale when you're just looking in the Hercules camera. You're just looking right what's directly next to you, and then when you peek in with the overhead bird's eye view of Atalanta, and you get a sense of the size of some of these things. Now that's some of these boulders and bigger slump failures. It's very big features, and
Oh, there's a
Sophanophore?
ctenophore.
Oh, ctenophore.
Well, and sat feed 3 is going to have the quad, so you'll get both views as well as 2 other views.
A viewer is asking, do the sediment samples ever get carbon dated to determine the age of the sediment?
I actually don't know of
Yeah,
of
normally,
dating sediments. Yeah.
yeah, I'm not familiar with that either. Normally when we take push cores with the ROV, the primary interest is the sort of sediment seawater interface. And in terms of dating, normally that happens more with the rocks, and in terms of sort of timescales, as Daniel was mentioning earlier, studying the sediment accumulation rates would be another kind of tool in a geologist's kit to study age in an area. But yeah, there's a lot that lives in in the sediment layer that we can't see, and so taking push cores is not only for geologists. A lot of microbiologists are interested in that interface between the sediment and the seawater and what's living there.
Looks like another acanthonis. Yep. So many. Yeah, they're everywhere.
So there's a question for Maikani. How did you get interested in ocean science, and what was your progression from college to your specialty?
Thanks for the question. So I kind of figured that I wanted to do marine science when I was still in high school. So when I graduated from high school and enrolled at Palau Community College, I majored in environmental marine science. But also I've been an intern at Pickrick since 2015 up until like I graduated with my bachelor's. So I guess that's kind of what helped me like gain experience and gain my love for doing ocean conservation and marine science. But yeah, I feel like
Yeah.
not most, but like people who live here and get to go out and see what's out there, that's what really helps drive their love for marine science. But yeah.
And then the second question was, what was your progression from college to your specialty?
Um, so after college, I did a 6 months internship with the Woods Hole Oceanographic Institution, where I got to work on different things. So first was coral ecology, and then the second one was doing deep sea studies in the deep sea in the Arctic, mainly looking at invertebrate larvae. And then after that, I kind of took a break for
Let's go
about
person.
a year.
I'm
And
gonna
then
push in on
Pickrick had
this
an opening. I applied, and here I am.
sea critter here. What is this?
Cucumber.
Oh. It's like in a different posture.
Mm-hmm.
Looks like a shoe.
It does look like a shoe.
Jake, okay to go cinema cam?
Yes.
Yeah, there's a little scale worm behind it. I think the scale worms follow these around.
This would be a pretty cool shoe design.
Yeah.
Yeah, I agree. How we get one?
I bet you could print one. If you like
Yeah.
your Crocs.
Uh-huh.
There's a lot of printed shoes these days.
I feel
Thanks,
like you'd
Jake.
have to print it, you know, it just to
it's
get the
gonna-
look right. Yeah, it's gonna swim.
Gonna take off.
Oh.
So this is a sea cucumber in the family Elpidiidae, probably an Amperima. Oh, and the scale worms hitching a ride. See their friends.
Yeah, those scale worms are quite beautiful. They're very vibrant colors.
Yeah, they're usually like a blue or purple. And I love their bristles. They're golden in color.
All right. Come on.
Have you noticed that the deep sea cucumbers don't look anything like cucumbers?
How'd they get the cucumber name?
Probably because the shallow ones kind of look more like cucumbers.
These are the deep sea shoes.
Yeah.
I remember there was a coral that was collected a few years ago and it smells like cucumber or something.
It smells like cucumber?
Yeah.
Oh.
That was very interesting. I can't remember.
There's also
I think I have a picture of it in my phone.
There's also a little fish in Alaska that smells exactly like a cucumber. I
Oh,
don't
that's
know what
strange.
species it is.
There's a spiky
I'll
Yeah.
look it up.
seafloor critter. Probably another cucumber.
Yeah, that looks like the one that we collected.
Yeah, it does look a lot like the one we collected.
Quick snap zoom there.
Okay, also on cinema cam.
Yes, all good.
Yeah, so this is in the same family as the one that we're just looking at, but it's
Excellent.
different genus. But it still has a scale worm friend.
All right.
So this is
Come
the
on.
family Elpidiidae. That one looks like more like the one we collected on, not this dive, but the previous dive.
Yeah.
So the one that smells like a cucumber in Alaska is called the rainbow smelt, but like locally it's known as the cucumber fish.
The cucumber
And it
fish.
smells exactly like a cucumber.
I never would have thought a smelt would smell like cucumber.
It's the strangest thing. Like
That is so
you
weird.
just get this giant whiff of cucumber.
Honestly, I think that's better than like fishy smell.
That's true.
You know what would be crazy if they put that fish in one of those like blind tasting competition. Like,
Just smell
smell
it.
a
What is it?
cucumber, eat a fish.
That'd be so mean.
Also, can we trigger a Niskin?
Oh, yeah.
Megan, what's our range to waypoint 3?
Oh, we're pretty close. Looks like 100 meters.
Roger, thank you.
Pan left. Perfect. Which Niskin are we going for? 2?
Yep.
2.
2.
Oh, there it goes.
Thank you.
So it looks like we've just now covered about 1 kilometer from where we started today's dive. Still have about 1.2 kilometers to the top of the summit. Also, the slope is going to start getting a little bit gentler.
Alright. Sarah, you want to fly? Alright, we're going to do a little pilot swap.
Copy that.
Give you a DVL reset real quick.
A viewer is asking, how cold is it inside this control room?
Too cold.
Very cold, especially back here.
We have to keep it cold for all the computer systems.
You're in full autos right now.
All right. But Jake's in a t-shirt.
Yeah.
Yeah, I don't understand that.
Crazy.
It keeps me sharp.
To be fair, I think May has the worst section. I think it's the coldest back there.
Yeah, it's really cold. I'm wearing 7 layers. Yeah, but I feel good if I wear all my layers and my hood. It's really blasting right behind us.
You
Like,
need one of those down suits, like full onesie down suits.
yeah,
That would be good.
maybe. It's just sometimes it's less cold, so I like having the option to be in 6 layers or 5. It's a good simulation for how cold it would be on the seafloor, though. That's why it's cold and dark in here, so we can feel more realistic environmental setting.
You're more connected then.
For our curious viewers, just under 2 degrees centigrade currently at a depth of 2,500 meters.
Try to get up ahead a little bit. Oh. Slow down.
Probably.
So if you were to take a temperature measurement on the way down, and as we do that continuously, there's, along with the video data, we have various environmental sensors on the ROV which
Can we look at
collect
that little brown spot on the left?
temperature, salinity, measured as conductivity, and various other environmental parameters. The top 100 meters or so is what's known as the mixed layer. That's where the temperature is constant, and the depth of that is a function of how much mixing there is, so how much wind there is, and so that those top 100 to 200 meters are typically uniform in temperature. After that, you have a rapid decrease in temperature with depth.
Have a little cucumber
That's
go
known as
flying
the
by.
thermocline.
Ah.
And then after that, you'll start having a pretty uniform temperature.
All right, going on.
Most,
Oh, might have just caused a
You'll be good.
little bit.
Let me tilt up for you a little bit.
Tilt down.
I'll tilt up a bit.
Oh,
There you go.
Sarah, okay to take a cinema cam close up?
Oh, it's getting a little dusted, but yes, I'll try and get up and do that for you.
That's
See-
good. Yeah, got one.
See if we can.
Looks like an enemy, probably in the family Actiniidae.
Nope.
Gauges,
Yeah,
but
he's just getting dusted out. We zoom in on that guy? Unless we are.
This is a Umbellula, a type of sea pen.
down here.
Yeah, Sarah, if it's all right, I'll try to do a zoom in here with Cinema Cam.
Yeah, absolutely. Try and turn a little bit for you.
That's great. Thanks, Sarah.
Yeah. All right, come out. Thank you.
Faster, go.
Get back ahead a little bit.
Yep, I'll come up.
Yeah, so our scientist ashore, Dr. Kenneth Sulak, from the retired now, but previously at the U.S. Geological Survey, providing here some interesting fact here. So we're about that large top predator that we saw. So just mentioning
See
that
that little
neither
stalk right ahead
the
next to the
Bathysaurus
lasers?
ferox or this conspecific, the other
Snap really
species
quick
in
on
that
that.
genus,
Yeah.
Bathysaurus, that they're not entirely immune to predation.
Zoom in.
There's some deep sea skates that are known to feed on these types of bottom fishes. So
It's
yeah,
like a tiny sponge.
thank you so
Yeah,
much, Ken, for that
stay
fact.
over here. Come on. Try to come down for a moment.
Yeah, I saw it. It was pretty good. That's good. Thank
Okay.
you.
Come out.
Also seeing some new faces here in the chat. We got Emily McLaughlin from the University of Alabama. Thank you so much for joining us. And also Valerie Brown from American Samoa. Talofa. Great to see you,
Talofa,
Val.
Val.
And that's a perfect segue maybe to introduce this relationship that exists between Palau National Marine Sanctuary and the National Marine Sanctuary of American Samoa. So both of these sanctuaries have a sister side relationship, have worked together in developing their management plans, and there have been staff exchanges between both sanctuaries. They have similar environments, similar kind of challenges, and have been working together. And then in fact, Nautilus was in American Samoa just a couple months ago, and then
Nice.
when we planned the expeditions to American Samoa as well as the one here to Palau, we worked together with staff from both sanctuaries, and it's been a very rewarding and enriching process, and great to see you, Val.
We're completing our collection of lizard fishes today.
Oh, is this another lizard?
Yeah. So this is the tripod
Scoot a
fish, the
little closer.
Bathypteroa.
How big do those get?
That's about as big as they get.
Oh, wow.
Sarah, okay to
Can you
take a zoom in?
on that? Yes.
Sarah, also okay with cinema cam?
Absolutely. Ooh. Turn the zoom a little bit. Turning, I guess. Sorry guys, stick lock didn't want to stay there. Let me see if I can reposition a little. Nope, it won't let me stay down. Sorry about that.
No, that's all right. Thank you for coming.
Back up
Yeah.
a little.
So you see how you're kind of facing the slopes like down a little bit? So
Yeah.
you're on the corner. So if you come around and you try to face the thing kind of going up the slope, it's a little bit
Yeah,
easier to
dust in
land.
it.
Yeah, we can go look at that other thing. There was a round.
Yeah, to the port. There he is.
It might just be a rock, but I don't know. This is
Only
very
one way to
round.
find out.
Or
Yep,
coconut.
only one way.
Yeah, hoping my little dust doesn't get over to it. Come down real easy. It is, it's almost to it. Will you zoom in?
Yeah, looks like a rock.
Looks
Yeah,
like
it's a rock. Ah, well, I was hoping for an urchin.
All right, come out.
So yeah, a question here in the chat. So we have not yet collected on a rock. We've been looking for them. There was one spot earlier in the dive where we saw some rocky terrain, but they all seem to be not in place volcanic rocks, but things that had come down likely from the top of the seamount. So hopefully as we keep climbing the seamount, we have about 280 more meters to the summit. Hopefully as we get closer to the top, we'll find some places where we'll find some
Fish right there.
some in-place basalt that we can sample. So yeah, we are looking for it.
You zoom in on him.
This is another Aldrovandia halosaur. Also seems to be quite common down here with the Acanthonus armatus.
So a question to our pilots. How long is your watch and what do you do when you're not piloting the ROVs?
It generally here we tend to do 4-hour shifts. We call them split shifts. So we'll do 4 hours AM and PM. They generally run 12 to 4, 4 to 8, and 8 to 12. So if I, for example, like right now I'm on the 12 to 4, so I'll do midnight to 4 AM and then noon until 4 PM. And then in the evenings, we also come back and relieve the 4 to 8 shift so that way they can sit and have dinner as well. In between, I tend to kind of get on a schedule and stick with it. Like my evenings after dinner, I go work out, shower, take notes, emails, work until the midnight shift. And then I generally sleep from like 4 AM until about 10 or 11. What about you, Jake?
Yeah, pretty much the same. Get sleep wherever you can, and it depends. Sometimes it'll be different when we're in port. We'll work just during the day and kind of go 8 to 10 hours from the morning through the afternoon. But then, yeah, once we're out at sea, we're on the split shifts. because we're doing 24-hour ops.
The rock.
There's a fish. That sort of black floating,
Little.
yeah.
Come down here. Oh, we zoom in on. Come on in. Oh.
Another tripod fish.
But yeah, every different department definitely has different off-watch tasks and different schedules. So
So in this scenario, what Dan would tell me is fly the vehicle, not the camera. He drilled that into my brain.
Looks like the off-watch activity of the science department is watching Nautilus live. We have several of our
Get that out of the way. There we go.
other scientists also watching right now and very active in the chat. And it is fascinating and captivating.
Yeah. Fly the vehicle, not the camera. I hear that in my mind.
Usually
all
I
the
forget
time.
about the cameras. That time I was like, oh,
Yeah.
the shadow of Hercules in the at Atlanta view looks like kind of a figure. I don't know.
Yeah, we can let this one go. It's just gonna keep swimming away from us.
yeah. He's settling in. Oh, maybe close up. As the dust comes up, zoom in. Nope, another one that I just keep.
Yeah, no, it's just, the fish is not gonna cooperate. We've seen it before, so we'll probably see another one. And we know what it is.
Maybe if we can take it. There's both.
Oh, that's the Parapaguris, but
By both of
what's
them, there's
the-
two.
yeah, what's that one? Yeah, the red.
Come down over here. Okay, try and lateral over.
I think it might be a hermit crab as well, but with an anemone.
Looks like it, yeah.
It's
Try
such
and
a
get
big
out in front of
anemone.
it.
We haven't seen any more of those big barrows. Kind of seeing if there's, I mean, there must be smaller ones, but they were both kind of, or all three of them almost identical in size.
Get a little closer. Go. Still wants to.
Yeah, you gotta kind of give it a little bit of a turn as you land if you're landing on the slope like that, along with the downward thrust.
Okay.
The other thing you can do is click in auto heading sometimes, and when you land like that, and then right before you land, you want to frame it up before you land, like so that whatever you're trying to zoom on is right below the lasers, because that's where the camera zooms on. So you think about it, the lasers are right above where the camera is, straight out. Yeah.
Is that a stick or another crevasse up in the?
All right, zoom in.
Looks like a stick.
Perfect.
Okay, also to do on cinema cam, we'll zoom in.
Yep. Okay.
What's a crab?
No way.
Wow. Its backpack got really big.
Winsbeth style for sure.
Yeah, this is like ultimate style.
Style
Full points.
He stopped moving.
That's good. Thank you.
Right. All right, come out. Oh, maybe we can.
Yeah, it's a stick.
Yeah. So this is going to be hard because you're looking down slope. Yeah. So the back end of the vehicle is parked. And it can be hard to hold position there, and then also you have to look further down with the camera.
Yeah.
So it makes getting zooms harder.
We try and zoom in. There. Oh, where'd my thing go?
Do you say you want me to zoom in?
Yes, if we can.
Okay.
See if I can get this down.
Yeah, so in this scenario, you're not, if the vehicle's moving, you're never going to turn left to get to it because it's going to keep moving. So you got to keep the vehicle stable first before moving the camera.
All right.
I'm going to zoom out.
Coming up. Yep. Come out. Thank you.
I'm going to come up on the winch.
Back up.
Mike's Mike's in there cleaning the grease.
Nice.
A viewer is asking what kind of tools is on the ROV.
We got all sorts of tools. We have 2 manipulator arms. One that's position controlled that we've been using for most of our sampling, and then the other one is a rate arm that's a little bit slower, but it's useful for many tasks like holding on to things and that sort of stuff. And then we also have a lot of hydraulic functions, like for our bio boxes, our rotary sampler jars. We have a suction hose that we use to vacuum up things. And we have a number of other sampling tools like scoop, and we had a net on previous dive. Yeah. Also push cores for collecting sediment samples, and knives. We have knives on the vehicle in case you need to cut something.
And then obviously a whole bunch of, for propulsion, various thrusters,
Yep.
various environmental sensors. So there's a CTD, a pressure sensor, an oxygen sensor. Currently not carrying it, but we also have a temperature probe that you could stick into things. There's also various sonars on the vehicle that helps for navigation to look at what's in front of you. Obviously the cameras,
Yep.
High definition cameras, standard definition cameras, and then a whole bunch of lights to also
14,
light up.
15 lights.
And yeah, it's a very customal, and we also currently have a multi-beam on there that we can turn on or off to map with the vehicle. And yeah, that's a little bit of a Swiss Army knife of a tool. Has all kinds of tools. When you are exploring that deep, you never know what you're going to find. So having a wide diversity of tools is good to capture those. Also has various sampling boxes that are able to deliver up to 250 pounds of samples to the surface into various compartments. So when we're sampling, you typically hear an interaction between our science manager or data logger with the nav team and the pilots to communicate in what sample box, what sample goes in. We try to keep those separate if need to, so we know exactly what was collected where. Those sample boxes are also sealed, so it brings the water from the depth where we collect the organism or the rock.
Starting to get out ahead a bit, Doug and Atalanta. So a good visual I use is the nav screen. The boxes are usually set to 10 meters.
Mm-hmm.
So you try to stay within 2 to sometimes 3 boxes.
Mm-hmm.
Stay in the box. And then also the Atalanta camera, call that staying in the box so you can see the vehicle. And then when you see the Atalanta camera turn away, that's when you know you're pulling on the tether.
Okay.
So for our team on shore, we have now passed waypoint 3. So we are headed further.
Trying trying to see if there's
southeast
anything to stop
up the
it.
slope of the seamount towards the summit. About 250 meters in vertical relief to go until we reach the summit. Just under a kilometer to go.
So when I get a little bit out ahead, a trick I'll do is just like lateral.
We'll have about 7 hours
sides.
more of bottom time. So
See a few little
lateral
things up here.
Yeah.
on this expedition and the planning of this expedition, we've been kind of wanted to strike a balance in terms of mapping features and then spending enough time with the ROVs so that we can get a representative cross-section of the habitat. Over the next, once we
Go in.
leave bottom tonight, there'll be a little bit of a longer transit, so about 13 hours
We zoom in on that.
transit and then some mapping. So the next dive is going to be tomorrow night, launching around 8 o'clock local time here in Palau.
Feel for holding or steady there.
Yeah, that's because the
Oh, do you think of
tether's
this
stretched
black
out.
coral?
Yeah.
Yeah, that's good. It is, and that's
You think it's
Yeah.
another, like a heteropathes, like the one that we saw previously on the other dive?
yeah, I think so. And that we sampled. I actually haven't checked it out in the lab.
Nice.
Should come have a look.
Yeah.
Got a lot of samples.
Nice. All right, come out. Thank you.
That's great.
Couple little things up ahead when we get there.
Well, we can stay and look at that coral if you want, since you've got some time.
Come.
Yeah, if that's all right.
Awesome. Yep.
Waiting on Atalanta to catch up.
Yep.
She's coming.
Yeah, that's a beautiful line up there.
Just gonna try and bring her down a little.
And is it okay to also on cinema cam do a zoom in?
Yep. All right. See if I can tilt up a little and zoom in here. Tilt. There we
That's
go.
great.
Go down a little. Oh, she's so quick.
Yeah, it moves quick when you're zoomed in.
What kind was this one?
It's a black coral called heteropathes.
I think that was my first somewhat successful landing.
Yeah, no, that's
that's
good. You
a
did
great
great.
Good
shot.
practice.
Yeah,
Alright,
that's great.
you got your
Thanks, Sarah.
pictures,
Yeah,
Daniel?
yeah, that's phenomenal. Thank you.
All right, let's go out. turn off. Yeah, we had a little something over here
Yeah.
as well.
I wonder if they're the same. Yeah, I think they might be the same, that same black coral.
Yeah, it looks like it.
Yeah. All right. Zoom in on that. I think it is too.
Is that a shrimp?
Yeah, it's good looking shrimp. All right, we can zoom out. Yeah, down in the lower. It's got some big antennas. What's that
Oh,
little
it's
guy?
a copepod.
Oh, yeah, there's this little shrimp guy there. There he is. Dusted him a little bit. Oh, oh, oh. Oh,
I like when they do that.
he's gone. All right, there's another one.
Yeah.
About the same.
Yeah, same thing.
Now, do they tend to kind of cluster together a little bit?
I don't know.
No,
Probably not. It
no.
just seems to be the place where they settled out.
Yeah, in somewhat shallower water in certain parts of the world, and including in Hawaii and in the Mediterranean, also in Indonesia, there's these massive forests of black coral, typically depths of about 200 to 500 meters, where in all directions, everything you see is just black coral, one next to one another. But yeah, this is in a different family that are found in quite a bit deeper. The Schizopathidae, depths about 1 to 5,000 meters, and those are much more sparse.
Oh, there's another one. Seems to be a lot around here.
Yeah, so black corals are a group of predominantly deep water corals, about 250 species found around the world in all oceans from as shallow as 5 meters to over 8,000 meter in depth. Their closest living relatives are the scleractinian corals or hard corals. So like hard corals, if you would look under very closely, you would see that they have polyps with 6 tentacles that are unbranched. Black corals get their name from their common name from the color of the skeleton, which is what's underneath the tissue. So when you, on those zoom-ins, you could see the tissues, which is the brown material that's surrounding the skeleton, much like us humans. The skeleton is underneath it and inside. Black corals have a proteinaceous skeleton, so that's different from your hard corals, which have a calcium carbonate skeleton. And they're also important in many cultures. pathies, that suffix that you have. Huh, what are we looking here? Oh, another one of these sponges.
Some long, what is this? Ctenophores? Or?
Oh yeah, there's the benthic ctenophores on them.
Zoom in on that.
Cinema cam might
The
get a good
ones with
shot
the
of it.
silent T.
Yeah, that's one.
So,
Okay to zoom in on cinema?
okay to zoom on cinema.
Oh, a little bit further up.
Yeah.
Close, though.
Tilt up. There we go.
Yeah, there's a lot of them.
Coming up on the winch.
Oh, and there's also the arcturid isopods on the stalk of the sponge. You see those little friends with their arms in the air. They're filter feeders, so they're trying to catch particles. There's like a whole gathering of them.
Start to move a little bit. All right, zoom out. Got a little fish around here, I can see.
Oh yeah, there's
There
a
he
tiny
is.
fish. It's a little rat tail fish.
There guy. Zoom in. See if I can. Maybe. Get back down. Oop. Startled him. Nope.
Yeah, they move fast. It wasn't going to stay in the same spot.
So check your camera. It might be pointed a little farther up, so it's harder to get closer to things. So if you tilt down,
Yeah.
you can see
Kind of
how far from the
get
port you are.
situated.
Yeah.
Yeah. Oh
And
yeah,
if you're
that's
left
pretty
or
high.
right, yeah. Nice.
And a little right. It's a little bit better. I see something off forward to the right, or to starboard. Go check that out.
Oh, well,
Not sure what
maybe
it
it
is.
wasn't cookies. Yeah, I guess not.
Looks like a cucumber.
Yeah,
Yeah.
definitely a cucumber.
The one that's like the sweet potato of
the
cucumbers.
sweet potato cucumbers. The one with the really nice texture in the weirdest way, or however I worded it.
I love that in Atalanta's camera you can actually see us like looking at it.
Yeah,
Oh,
it's a big cucumber.
did not.
Kind of looks like a loaf, like maybe
Yeah.
one of those roll cakes.
Okay to zoom on cinema?
All right, yep, go for zoom.
Nice.
You can zoom in. Reach to you.
That's great. Thank you, Sarah.
Yeah. No. Yep.
Cool thing about sea cucumbers is they're echinoderms, and echinoderm means spiny skin. And so when you think of echinoderms, you're often thinking about sea stars and sea urchins, and these things have spikes and spines all over them, right?
Very cool.
But
Well,
the sea cucumber looks really smooth. But if you look very, very closely inside their skin, they have these things called ossicles, and they're like little spines in the skin. And each species of sea cucumber has a very unique set of ossicles, kind of like the spicules in a sponge. And you can use those to help identify species of sea cucumber.
Oh. Oh no.
Just doing a pivot for us.
Yeah. I don't think I startled them enough. My goal was not to. Excellent. I think they're so pretty. Oh no, he's gonna go. I think we'll get going. It's like one of my favorite things when I come back home and stuff and people ask me like, what's some of the coolest things you see? And I'm always like, oh, these cucumbers, when they get upset and they like
no one
undulate in front of us. No.
They're like,
so
what?
beautiful. Ooh, we got a fish. Maybe. Or psych. Nope, it is.
It's quick fish.
Yep.
This is a cusk eel, a fish in the family Ophidiidae. So don't be confused. It's not actually an eel. It just has sort of a long tapered eel-like body, and so that common name eel comes about. This one looks like it's another Acanthonus armatus. It's got that really big head with the small eyes. Fun fact about this fish, it has the smallest brain to body size ratio of any vertebrate. But it knows that the ROV might be a bit scary.
Yep. Try to gently creep up on it, but...
Okay to zoom on cinema?
Yes, okay to zoom.
Did you want me to zoom as well? I can't
Wonderful,
tell who you're
thank you.
talking to now.
Ooh, there he goes.
Let's go.
Yeah, the long tapered body form makes for more efficient swimming in these animals, but they tend to move a bit slower than your surface fishes.
Yeah, if he's just going along like, I know you're there.
Yeah.
Oh.
So just as a quick time check, we have a little under 7 hours of bottom time to go. We'll be leaving the seafloor at the shallowest portion of this seamount, which is around
I just really appreciate
2,200 meters.
how large, like...
Should take us about 80 minutes to reach the surface, so...
Yeah,
We'll
they
be,
do. You know, for a fish with such a large head, you'd think it would have a bigger brain.
be securing the ROVs on deck around midnight, and then making our long transit, a little over 130 nautical miles to our southwest to go closer to another seamount. This one is south of Angar Island, also within the Palau National Marine Sanctuary. And that seamount is also very interesting. It's actually located along a very long ridge feature that has not yet been mapped, and so we'll be mapping that for a while to try to pick our next target. That dive is likely going to be somewhat shallower than this. So we've had some nice depth range, but that seamount looks like it's about 1,500 meters at the summit, so quite a bit shallower than this feature. As we're here starting to prepare for a watch change, it's been a very interesting watch, and collected our Niskin samples. Came across some very interesting slump failures, some really interesting burrows. Collected a shrimp sample, a cucumber, and documented a whole bunch of different fish. The sediment has been continuously heavily sedimented. We've just seen a few rocks here and there.
Hello, hello. We're currently doing a watch change, so it'll just be a couple of minutes to settle down.
So did I understand that we're not in orbiting? I would be in favor before we get the ship moving while we're popping up and doing an orbit sweep and seeing if there's anything in our future.
Yeah, let's find some rocks.
Roger. Let's see if it breaks our WCA cameras.
Atalanta's out in front of Herc. You want me to do anything about that?
Come up, come up, come up.
Well, good afternoon, good morning, everyone. This is the 4 to 8 watch just getting settled in. Give us another minute to get all of our screens and laptops configured, and we'll start introductions of the watch in just a moment.
Oh, I see what's happened. That camera is... What? What is going on here? Huh. Can you, when you get a chance, put a note in the red book? It looks like... that camera is crooked.
Which one?
Upper. Oh, you'll have to come up faster, faster, faster.
Copy. Yeah.
I need to go back down. We're late. You ought to be able to see for days from there, Chris. 35 meters up. If you look straight down there, Jaden, I'll come under you and we'll see, make sure the tether's not.
Copy.
We can, Al, we can do introductions whenever you want.
All right, Ali, and good afternoon. It is 4:04 Palau time. And while our navigator and pilots get situated, I'm going to ask the back row to do some quick introductions.
Thank you, Al. Good afternoon, Ali, everyone. My name is Brian Kennedy. I am the Chief Scientist at the Ocean Discovery League and the External Science Advisor for NOAA's Beyond the Blue campaign. I'm a deep sea benthic ecologist by training, and I am the 4 to 8 watch lead.
Ali, everyone. My name is Harold Carlson. I'm a PhD student at the University of Hawaii at Manoa, studying oceanography and deep sea ecology, and I'm in the science chair for the 4 to 8 watch.
Okay, you can look up a little for us, Jade.
Aline, hello everyone. I am Ayla, and I'm a Marine Lakes Research Assistant at the Coral Reef Research Foundation, and I'll be the data logger for the 4 to 8 watch.
And I'm former Congressman John Culberson, and active supporter of the sciences, and especially ocean exploration, space exploration. In my time in the Congress, I served 18 years representing West Houston and served as the chairman of the Commerce, Justice, Science Appropriations Subcommittee. And it was a great pleasure for me to be able to help over the years make this extraordinary exploration like this one on the Nautilus
up higher
happen.
You're crazy. You're crazy. Here we go. Coming up. Roger. Okay, Jaden, you got to come up with me.
Copy.
Yeah, we'll keep that 24 there. Or 25-ish delta.
I just gotta say, John, I'm always glad that you're on our shift with us.
I enjoyed it immensely and try to help any way I can to I am an amateur scientist, amateur oceanographer, astronomer, and it's really a joy for me to be out here and be a part of this exploration of parts of the ocean and the world that no one has ever seen before, and the discovery of species that no one has ever known about before.
It's 40 meters there.
Yeah, thank you.
Out of beams. Spinny spin.
Hey Brian, is it possible that shipwrecks, entire ships could be buried under sediment? Is that a possibility?
Alexa, spin Herc slower.
In the grand scheme of things, it is possible.
Let me do that.
But sedimentation rates in the deep sea are exceedingly slow.
Max velocity, we'll set that to,
so
what do we do
it would
this
have to
before?
take, would take like a landslide or something like that to fully bury one. But in places where rivers and stuff flow out, where there's a lot higher sedimentation rate, they can be, collect a lot more sediment. Out here, not so much. So
it's as slow as it goes. Alexa, spin Herc 180 degrees starboard.
So what we're doing now is we've been really seeing a lot of sediment on this dive, and so we've got this fancy new multi-beam on Hercules. And so what we've done is we popped up about 40 meters off the seafloor, and we're going to take an extremely slow 180 degree spin to allow the multi-beam to look out into the abyss and see if there's any features out there that might be hard grounds or something like that that might give us a little more diversity of substrate. And so we'll be up in the water column for just another minute or two while we spin around and use the remote sensing of the Norbit multi-beam to see out into the abyss. And if we find anything, we'll head that way.
Brian, that was a good call for y'all to do that. Remember yesterday, of course, the Norbit side scan sonar identified two bright objects that were tall, looked like pillars. And as we drove over there at the end of the dive, discovered this really large rock that looked like Grand Central Station in New York City. There was so many animals living on it. So it's a great idea to get up and take a look out over this featureless mud plain to see what's sticking up. What is the range? How far out can the radar sonar see?
You know what I did? I'm really silly. I spun the wrong way. I'll let it spin on around, then I'll...
Chris, what kind of range are we getting?
Oh, what's the range, Chris? You need to turn on your mic.
Sorry, I had SPL off.
Yeah, how far out can you see?
It's about 70 meters, 80 meters out to either side, so maybe it makes when we do a spin like this, it maybe makes, I don't know, 150, 160 meter diameter circle.
We're seeing a few individual dots there. What are those? Some are disappearing. Are they artifacts or?
So right here, that looks like something hard. There's a small rock here. I don't know. There's honestly not a whole lot. That's super interesting.
We need to come up with a different, an all-seeing eye nickname there.
So you're facing north now? Yeah.
Yeah, but I'm going to take it out of
yeah.
Auto Magic here and take the turn back out of my tether and do some little wash machine action here because we got...
Hey, Amber, what PC on the router is the Norbit?
So I put it on PC4.
You can see it.
You can also see it on Herc Rosproc.
There's a Rosproc button on your panel.
Okay.
Yeah, you should be able to see it.
And the Norbit is on?
It is, yeah.
Terrific. I'd love to see it. The Norbit's the one that's a really high resolution. It was giving us those extraordinary images yesterday evening.
So just to update our viewers, we are in the Palau National Marine Sanctuary.
Can you see it from there, John?
We are...
Okay. So we have a couple
So we are surveying an unnamed seamount, which was only partially mapped prior to the EV Nautilus, acquiring the remaining coverage data this morning. This seamount summits at a depth of 2,200 meters.
And then there's something. The first target is just to your right. Maybe.
We are currently 60 nautical miles southeast of Bubble Daub Island.
You can move the boat.
And we are at a depth of 2,359 meters.
East, northeast. Give me a cardinal point.
Sorry. It's east.
Roger.
Bridge nav, 15 meters, 160.
East is right on the nav screen, so you can also say right, and it'll actually go the right direction.
So wait, are you north up on
this is
your display?
Now it's north up view. So it's actually from where you moved a bit. So it's
you're going for the little one. Okay.
Well, I'm going to go there first, and then
got it. I understand now.
Well, I can fly over it at like 25 meters. Or you want to come right down?
No, let's go down and see it. No, let's go see it.
Yeah, you can see that it's that bright purple mark on there.
Oh, I'll just fly to that.
Yeah.
Why don't you just tell me fly to the bright purple mark?
So can this multi-beam of see kind of see like animals, or is it just mainly boulders, rocks?
At the right angle, and you could see some animals at this altitude, and down looking, probably not. We're probably going to be looking for rocks or hard substrate, but if we spun the transducer around and looked in front of it at a close range, yeah, you can definitely be able to image
you're still up in the water column.
Some animals. Yeah. Generally animals with harder skeletons and stuff like that. They're something that you can get an acoustic return off of.
Like the corals last night. We could see the corals on top of that big rock.
So you can actually see fish very well in the water column data on this.
Sorry.
Most fish have swim bladders, so that...
Not at this depth, though.
Not at this depth? No.
Yeah.
Well, darn.
And bubbles are very...
I see schools of fish and stuff in it all the time on the surface. It's really cool.
Sure. That makes perfect sense.
And for our viewers, we have that Norbit image up on SAT-3.
So we need a suggestion. To me, that looks like an eye, but the eye of what?
Wow.
Needs to be some mythical.
I mean, we were all thinking there.
Is Sauron would work.
I think Eye of Norbit is better.
The Eye of Norbit.
That works. That works.
That works.
The Eye of Norbit.
The Eye of Norbit. The all-seeing Eye of Norbit.
Yeah. That's good, Brian. That works. That's it.
I think another 5 down will do me. And if you want, you could spin.
It should be kind of off your aft quarter, aft starboard quarter.
If you, Roger. If you want, Jaden, yeah, you're already on it.
This one might be nothing. It was pretty. I just wanted to check because we were so close.
Well, that is different.
Is this the smaller of the two objects, Chris, that you saw?
Yeah, yeah. I think you're directly over top of it, so back up a bit.
Is that a crab?
Yeah, it looks like a squat lobster, and there's a rock. Well, we proved it can find a rock.
This is what it saw directly under us.
Yeah. There is a squat lobster, it looks like, a little bit ahead of us.
Was there some, like, behind you? See behind on the Atalanta view?
Oh, yeah.
I think that's maybe what we saw. Or no, that's just your shadow.
That's the shadow.
Yeah.
Yeah, we saw our shadow, that's for sure.
Okay, so, well, we did find...
6 more weeks of winter.
Well, that gives you the idea of what we can see with it.
Yeah, that's pretty good. So let's take a quick look at the rock, but I definitely want to look at the white crustacean in the sediment just on the far side of the rock.
All right, Roger. Okay, I gotta tweak
another
my dials
squat
back
lobster
here.
there.
Yeah, it looks like...
Is this a rock or is this wood?
Yeah, I don't know yet.
Yeah. Looks like maybe wood.
Looking more and more like wood the closer we get.
Come down 5 for me.
Come down.
Can you hand me the sample spread? Hey, can I see the sample sheet for a second?
Unfortunately, I don't have a sense of which way is uphill at the moment.
To your right.
I know.
Actually, you're more or less pointed uphill.
I have to disagree. Unless I set Herc down on a rock.
There's uphill that way, and you're pointed straight uphill.
Let's see where I just landed. That's tail of Herc there landed. Maybe not. I couldn't get the front of the vehicle down.
And the ship is headed almost due west, is that correct?
The general
It's pointed due west, but we're moving south.
Maybe it's just that soft.
Yeah, I would expect the sediment to be exceedingly soft and clayey.
Yeah. Do you want a close-up of the wood? Or?
Yes, please. And there's a distinct possibility we're going to want to collect the squat lobster in the wood.
What's that?
And there's a distinct possibility we're going to want to slurp the squat lobster sitting in the wood.
The one in the wood?
Or I think they're likely the same, is my guess.
I'll come back around to that angle and see if I can completely destroy the site.
A bit smaller.
Get a look.
Probably more slurpable.
Exactly. The earlier wood fault we saw had multiples of these white immunida living on it. And so I'm assuming they're all the same thing.
Push in about halfway for me, Amber.
Copy.
So,
yep, there's some of those same urchins we saw earlier and that we collected a couple dives ago in the leaf litter.
So we picked up something here.
Bubbles.
But I'm 90% sure that that's noise. But
turn on the downlights for me.
Or you found a methane seep.
No, or it could have been.
Downlights are on.
Yeah, or it could have been something that we picked up as we got lower and shot a more oblique.
Am I over the squatty there, or is it?
It was on the.
It's a bit further down.
It was on the on the closer end of it, but I now can't see it. It may have sought refuge further up the thing. Couple tube worms, couple of anemones. All right, let's go look at the big squat lobster just ahead then.
Yeah, I wonder if some of the animals we see hanging out on this plant debris have gut microbiomes that allow them to digest cellulose and plant matter.
I would not be at all surprised.
I don't think I've ever seen a squat lobster this large before.
It's
Are you sure the downlights are on?
That's a big one.
It says they are. I can cycle them.
And a hermit crab off to the left.
That's all right.
Oh, and it looked like a decorator.
The starboard port lights are off if you want those on.
No. I think we have some light issues. You can make a note in the red book. Check the lights when we're back on deck and stuff. So. Okay, Amber, go ahead.
Zooming in.
Yep, this looks like it's in the Munidopsidae.
Yep. If that is slurp sizable, or size for slurping, I would like to try it.
And what's the common name for that, Harold?
Squat lobster.
There he is.
And
we can also try and grab it with the arm if you prefer.
Try the net.
I mean, we also have the net. We could gently scoop it up. I think the net is a bit fragile at this point, so we'll have to be careful. But
it was fragile to start with.
It was fragile to start with. That's right.
There is a small chance, so but then they'll get back out. We grab them with the slurp, and that, gets the kid's toy stuck into the end of the vacuum.
Yep.
And then we put him in the box, but he's just going to swim right back out of the box. So
I will defer to your expertise as the manipulator arm operator. What do you think is the most effective way to capture the squat lobster.
Yeah, so he's obviously at 10 centimeters or 4 inches too big to go in the slurp.
Yep.
And if I wound him, he would not be able to swim away, but chances of grabbing him are
limited.
Limited. Yeah, I think the net is probably the best option, but then that burns the net, so.
Well, that's probably okay.
Right.
It would just be great to see the net get any use.
We can also we've seen 2 or 3 in this dive, and if this is an impractical collection, we can also keep our eye out for another one and not.
I'm willing to give it a go with the net.
All right, let's give it a go with the net.
Okay, see if I can get the net without carrying it away.
Is this a normal size for a squat lobster?
This is huge.
Oh.
This is a very big one.
Okay, we got a viewer said this is a chunky squat lobster.
Well, we're always open to common name suggestions, so.
And has this species lost its coloring and become pure white because of the absence of light?
It is a distinct possibility. That's a hard question. We're really just beginning to understand the range of how light and bioluminescence works at this deep. There's obviously no light from the sun this depth, or this deep under the ocean, but there are still lots of light-producing organisms, and we still see a wide range of colors in the deep sea, from whites to reds to pinks to purples, and it is. And I guess that answers that question.
That was a cute swim.
He felt just that slight vibration in me.
Yep.
That was it.
Wow.
Bye-bye.
I would say leave the net attached right now, because if it was that sensitive, I don't think we have a prayer at catching it.
I think we do with the net.
Okay.
But I'll have to have the net ready and then just put the net behind him. How do we catch crawfish, John?
You put a net right behind them.
Roger that.
Because they can only swim one way.
Is that right?
Yeah, they're not too bright.
But they typically put it behind. That's the technique.
Yeah, that's their, just like shrimp, that's their primary escape mechanism. They use that big, powerful tail to shoot backwards.
They do. We've also caught crawdads in Texas tying a piece of bacon to a string and dropping it down their hole. And they won't turn loose of it.
I used to catch them in San Diego like that as well as a kid. Take us all day to fill a 5-gallon bucket. First went down south. I was just amazed that you could get a 50-pound in fraction of the time.
They will not turn loose of that bacon.
No. Great fun.
Can't say I've ever tried this.
And tasty.
They are.
Just going to float up a bit here and see if we can spot it. Where'd it go?
You also learn over the years, don't ever eat a crawfish that's tail is sticking straight out. If they serve it to you and the tail's sticking out, don't eat it. That means it was not alive when they dropped it in the boiling water. Hard on a crawfish, but at least he's fresh if he's curled up.
I like watching seafood boils, so I've heard about that, but we don't have crawfish here, so I've never tried.
What kind of seafood do you boil that you serve here at Palau?
Mangrove crab, land crab. We got lobster too.
What's your favorite?
I gotta say the land crab. It's small, but you have to put in a lot of work to get the meat.
Yeah.
Yeah, but what about you, Ayla? What's your favorite?
I like the mangrove crab.
Okay, mangrove crab is a little bit bigger, so there's a lot more meat.
Yeah.
You're going to have to...
They're living off mangoes.
I see what's happening.
Mangroves.
Mangrove, I
Yeah,
see. Mangrove crab.
I'm at the end of my teeth there.
We even have mangrove clams that are pretty
kind
of famous for cook. If you eat it, you can eat it raw, or you cook it
What's
in
that?
like coconut
You
milk.
want me to turn so you have a
And
little
Cook them in
extra?
coconut milk. That
Yeah.
sounds
Oh,
wonderful.
I was just feeling the bounce of the ship there. It was too far away.
Do you have oysters here in Palau?
No, we don't have oysters. We have different types of like seagrass and mangrove clams,
Mangrove
but nothing.
clams.
Yeah, nothing like oyster.
Yeah, clams are good. They're delicious. They love them up in the Boston area and on the east coast of the United States.
We got giant clams as well.
What about scallops?
No, we don't. I don't think we have that.
Okay.
Yeah.
Or abalone?
No.
There are some scallops on the reef, but they're not like scallops like you eat.
And abalones too.
Yeah, they're also like
Push in there, Amber.
like a shell on the rocks.
Right. Oh, they're attached to the rock.
Yeah, but they're kind of tiny.
Negative.